Welcome to Chapter 6. This is Energy Flow in the Life of the Cell, and I'm Colin McLeod. How are these the same? So on the left, we have a marathon being run, and then on the right, we have, I think that is NASCAR. It is, at any rate, a race. Well, they're the same in that in both cases, we're basically taking one kind of energy, that is chemical energy. In the case of the runners, that chemical energy is stored in their muscles, specifically as glycogen, which is a polymer. It's converted to glucose, and then that glucose is used to make ATP, which enables them to run by various other mechanisms. The cars are similar. Instead of glycogen as their chemical store of energy, they have gas. And so they're both using chemical energy to do something, and that something is called work. And that's one of the main topics of the first part of this lecture. Energy is used to do work. So what is energy? As I said, energy is the capacity to do any kind of work, whether it's running, whether it is using the energy in fossil fuels to drive a car or a truck. That's energy, and it can be used to do work. Fun little thought here. Can heat be used to do work? I'm going to think about that one. We'll come back to that one later on in this lecture. Can heat be used to do work? Sometimes the answer is yes, sometimes no. Work is a force acting on an object that causes the object to move. So the man lifting or pushing this tire is a pretty obvious example. However, we have also learned some more subtle examples, such as moving a vesicle around in a cell, that is energy being used to move a vesicle. Even active transport, that would be the same thing. Energy is used, in this case ATP, to pump a molecule up its concentration gradient. And that also does require that protein pump do a little bit of shifting and moving. That too is using energy to get work done, even if it is only at the molecular scale. Energy and work in living things. This diagram is not in your book, but I thought I'd give you a brief overview of how living things use energy. So this circle I've used to describe a cell. So the inside of a cell, or you can imagine as just sort of the circle representing a whole animal, we intake food. So I'm not including plants in this discussion here. We eat food. We eat it because it has energy in it. Right away, we're going to have to process it in a couple of ways. We can store it as a short-term glucose, glycogen, long-term fat, very long-term. Glycogen is about 6 hours. And so we have these energy stores that we get from food. We all know that if we stop eating, eventually we will die. So we need food for energy, and we store it. We store it so that at a moment's notice, we need to run away from something, or we forgot our wallet at a bus stop. We got to run back there. Then we can take this chemical energy and convert it to ATP. For now, again, think of ATP as the money of the cell world. If you've got a lot of ATP, you can basically make anything happen. It's a corrupt cellular world. You can bribe the guy at the gate, everything. You know, it's a corrupt cellular world. ATP is money. You can get anything done with ATP. So ATP can be used to do work. Now, the thing is that we always are going to produce some type of waste product. The most common waste products in human beings and animals, but for most things really, is number one, CO2. That's why we exhale. We're getting rid of excess CO2. And then also heat. Heat is another waste product. And so think about that again, that question. Can we use heat to get things done? You might think of like a steam-powered engine. Oh, that's kind of like using heat. You could probably think of some engineering examples, but in terms of living cells, this heat is essentially considered waste. You cannot eat heat. You can't do much with it, not really. So types of energy. Firstly, there's potential energy. Secondly, there's kinetic energy. Potential energy is stored energy, stored in some way. Could be chemical, could be you're sitting on top of a hill on your bicycle, could be other things. Kinetic energy is the energy of movement. So let's take a look down here at this sort of representation of a pendulum. Now let's look first at the purple bar that goes up. That's potential energy. So notice This pendulum, the weight at the bottom of it has the most potential energy when it is at either extreme of its swing. On the left, on the right, that's when it has the most potential energy. So now that we've paused it right here, at this point, the the pendulum has zero kinetic energy because it is not moving. As it begins to move back this way, its potential energy will be converted to kinetic energy. And you'll see the potential energy bar go down and the kinetic energy bar go up until the maximum kinetic energy is attained right when the pendulum gets to the bottom. When it's at the bottom and starts moving up again, that kinetic energy is then again converted to potential energy, which will, and then when it gets to the opposite end of its swing over here, all the kinetic energy will be converted to potential energy. You can replay the portion where it was swinging and look at those bars going up and down again if you like. Now let's take a look at the bicyclist, upper right hand corner, and we're gonna ignore this first section of the diagram. So step one here, this bicyclist has potential energy simply based on its position on the earth and the fact that gravity exists. Things tend to fall towards large objects like a bicyclist. If this bicyclist just gets a nudge moving down the hill, the bicyclist himself or herself will not have to spend any energy because they have stored energy, and that stored energy is potential energy. And as soon as they start rolling, that potential energy begins to get converted into kinetic energy, and that will propel them down the hill. Now, to extend this a little bit into some more abstract thinking, it's a wild thing to think that potential energy just is merely where you are in relationship to the earth. You're on a hill, so you have potential energy. That energy can be converted into something, say, like a battery. If you're in one of those hybrid cars that when they go downhill, they take some of that potential energy and recharge their battery. It's pretty amazing that simply your position on a hill can be converted into electrical energy, especially once we begin our very simple discussion of thermodynamics. One more thing to say about kinetic energy. So kinetic energy, the bicyclist moving down the hill. This is not just for large physical objects that we can see. This would also be true for molecules moving around. So a molecule itself has kinetic energy. And later on, we're going to see that a certain amount of kinetic energy, oh, there's a camera, is required sometimes to make reactions happen. Like they must run into each other hard enough, and then maybe one of them will be broken or changed or split into two or something like that. Chemical energy. Chemical energy is a very important type of potential energy, and there are lots of examples. So food is a type of potential energy in the form of chemical energy. You can eat it and then use it to go on a bike ride. This is just a molecular form of gasoline. You can burn it to produce heat. You can burn it to get work done, i.e. drive a car. This is potential energy. Again, chemical energy stored in wood. You can convert it to heat your house. And electricity is also an example of chemical energy. We basically just have positives and negatives separated, and then when we connect them, the negatives will flow towards the positive, and that can get work done if you hook the battery up to something, like a light bulb. The laws of thermodynamics. Do not be intimidated by such a long word. It's not too complicated. Number one, the laws of thermodynamics describe the availability and usefulness of energy. So availability, again, think about that heat. How available is heat to use by a living thing? Can you use it to do something? You can use it to warm yourself up, but can you use it to fight a disease that has gotten into you? Maybe not. And then usefulness of energy, essentially the same thing. So number one, first law. Energy can neither be created nor destroyed. If you ever feel like you're losing an argument, just say that, and all of a sudden people will think you're very smart. So it can neither be created nor destroyed. So the energy in gasoline, if we just took, this is a simple math example, 100 units, some kind of units of chemical energy, and we combust all the gasoline in an engine, and 80 of these units are lost as heat energy, then we say that the remainder is used to actually do work, to move the car forward. And so again, this heat energy, even for a car, it's gone. You can't get it back. It's not useful. So again, how available and useful is the energy? That's what thermodynamics is about. So here, no energy was created nor destroyed. The 100 units in the gas, 80 of it went into heat, 20 of it went into moving the car. So none of the energy was destroyed. It simply changed forms. Heat is a measure of how fast molecules are moving. So the molecules in the exhaust and other parts of the car that got heated up, they would be moving faster. The other kind of energy is kinetic energy. The car is moving now. Okay, last point here, that's this little dash mark. The total amount of energy within a given system remains constant unless energy is added or removed from the system. What do we mean by system? System, it could be any system you decide. For this this PPT right here, I've decided that this system is just the gas, the heat, and the car. That's my system. So in that system, energy is going to remain constant. And that is sort of a matter of words. You could talk about the Earth having a constant amount of energy. Ugh, don't get too abstract with that, and we wind up confusing ourselves. Second law of thermodynamics, also not to be intimidated by it. The amount of useful energy decreases when energy is converted from one form to another. Think of the last example. You burn gasoline, you get the car moving, and that kinetic energy might be usable. If you're driving a hybrid, you could recharge the battery perhaps, but that heat energy is lost. So the amount, anytime we use energy, we convert energy from one form to another, and some of it is typically, not always, but typically lost as heat, and it's just not that useful. Arrow number 2, entropy. Also a very smart sounding word, or the disorder of a system tends to always increase. Disorder increases. If you look at how I kept my room as a teenager, that would simply stand as hard scientific evidence that order decreases over time. That's a joke. That would not be evidence. Okay, bad joke? Sorry. What this means in science is in a practical sense is that unless we constantly spend time and energy trying to keep things organized and ordered, like my room as a teenager, they have a natural tendency to fall apart and become more disordered. So, for example, what happens to you if you stop spending energy to keep your cells and body maintained and organized? Well, eventually you will die. The cell membrane will not remain as it needs to be. Concentrations of chemicals inside will gradually, as we know, diffuse outward and be unsuitable for sustaining life. Entropy increases. So take a look at just the top part of this picture. Ignore the bricks for now, just the top part. Here's a question for you. If the particles represent gas molecules at normal temperatures inside a closed container, which of the illustrated configurations came first? You can pause the video now and try to guess which one, and then explain why. More importantly, why. Okay, if you have not paused the video, last chance, now I'm going to explain it to you. Which one of these situations must have come first? It is the one on the right. This one must have come first because the second law of thermodynamics, when we convert one form of energy to another, and as time goes by, entropy increases. So disorder pretty much always increases unless we're putting energy into the system. So first, this state must have existed somehow. Who knows how? Perhaps a liquid was converted to a gas, and it was all in this region. Over time, disorder increases, and those molecules will spread out until equilibrium is achieved. The bricks on the bottom, we'll consider that example now. If you tossed bricks off a truck, which kind of pile of bricks would you be more likely to just accidentally produce? Well, I wish that I could just toss bricks off a truck. I need to build a short wall in my yard soon, and that they would just automatically land like the bricks on the left. Except that's not how it works. You have to put energy into making a wall like the brick wall on the left. Disorder is simply more probable than order. Entropy in action. The example, that's not my bedroom as I was a child, but the diagram on the left represents somebody putting a lot of energy into keeping their bedroom clean. There is less entropy or disorder. But disorder happens spontaneously. So on the right, as time goes by, if you don't put energy into organizing the things in your room, uh, randomly throwing your clothes around will result in chaos. That you will not randomly throw them into a folded position inside of the correct drawer. It just won't happen. So energy and work and living things. Back to this diagram. If we're considering an animal like a person, and they have eaten food, it could be used as or stored as glucose, glycogen, or fat. Here's a question for you. If a person eats 2,000 calories of energy in a day, gets 400 calories of energy of work done, and stores 400 calories of energy, how much energy is lost as heat? You can pause the video, reread it, and think about it. Okay, if you have not paused it, I'll now give you the answer. The answer is 1,200 calories of heat would be produced, because I've already told you that, what was it? 400 calories of work done, so work equals 400. They have stored 400 calories of energy, perhaps as fat, we'll say, and the intake was 2,000. So 2,000 minus 400 minus 400, the remainder would be lost as heat. 12,000 calories lost as heat. Another question for you. Which arrow is there the greatest increase in entropy? You can pause the video. Okay, if you haven't paused the video, I should suggest that you do now, because now I'm going to answer it. You should think about it. So what is entropy? All of these molecules, let's take a look right here. These represent potential energy in that they have stored energy. The food also has some stored energy. And what's going on when we take food in and then basically repackage it into molecules that are more useful for us, we're reorganizing chemicals. We're not breaking them down into their individual atoms and rebuilding them. We're more like breaking polymers into monomers. Glucose would be a monomer. And sometimes we're storing them as other things like glycogen and fat. By the way, I didn't expect you to be able to answer this question with this much articulation, more just for your critical thinking practice. On the right here, what's going on here is all of these organic compounds, remember they are carbon-based compounds. Glucose, glycogen, fat, they're all have carbon as the backbone of their structure. And that's why carbon dioxide is always produced when we respire them aerobically. We always produce CO2. We combine oxygen with these carbon compounds. CO2 comes out here. This is a huge, let me indicate what I'm talking about. Here, this arrow on the right, this is a huge increase in entropy because we're getting all these little molecules that are spreading out, diffusing out in all kinds of directions. Then you're exhaling them, they're going into the air. And also heat is a very unuseful form of energy. So we call it an increase in entropy when heat is produced generally because it cannot be recaptured. It's not organized in any usable way. These food molecules here, they have energy stored in their bonds in a way that is usable. To further explain what I was talking about on the last slide, let's look at the example of the respiration of glucose. This is the main way that cells use energy all over the body. In fact, the brain can only use glucose. You don't have blood sugar, that glucose, the brain goes downhill real fast. So let's look on the left. On the left side, this is where there is less entropy. There's less entropy. So why is it that there's less entropy on the left? The first thing that you should look for is, well, let's look at what's on the right. And the first clue is heat energy. Usually when there is heat being produced and released, that is an increase in chaos because that heat energy goes away. It literally shoots off of the planet Earth as infrared radiation into space and is never heard from again, gone forever. Um, that was a little bit into physics. My apologies. On the left, we have a total of 7 molecules. So that is 1 molecule of glucose right here. This is glucose, 1 molecule. And then right here, this is oxygen, and this big number here, the 6, that means that there is 6 molecules of oxygen. I won't be testing on this because this is more chemistry than you need to know at this point, just explaining it to you. So 7 molecules on the left, rather ordered. All of the energy that's really useful for a living thing is in just a single molecule, glucose, usable energy. Now on the right, we have 12 molecules and we have heat energy, so way more entropy and randomness. 12 molecules are going to spread out in all directions. It's like, it's like on the right, you're like trying to herd cats. And on the left, you just have a big cow tied to a post. They can't go anywhere. So on the left, on the right, 6 molecules of carbon dioxide and 6 molecules of water. So 12 molecules, they're running around like crazy. And so that in and of itself, there's more molecules, represents an increase in chaos or entropy or randomness. Heat energy is the kind of the final thing. That's why there's more entropy on the right. So using living things need to use energy to maintain homeostasis, to maintain balance in their cells, to get work done, to keep themselves alive. So the energy that ultimately sustains all of us comes from sunlight. So since all things, including cells, have a natural tendency towards entropy and falling apart, living things must gain external energy in order to counteract the increase of their entropy. Photosynthetic organisms, like plants, right down here, like plants, get their external energy from the sun. So that input of energy enables them to create order inside of their cells, specifically by doing photosynthesis. I'll give you that equation later and explain it. Non-photosynthetic organisms, like us, that is me on the right, in 1983, I have found my first tomatoes, first ones I ever picked. Like us, they get external energy by consuming the energy-rich molecules of other organisms. So little Mr. McCloud here on the right is going to eat those tomatoes, and the energy from those tomatoes will help all the cells of his body maintain homeostasis. The energy from the tomatoes will be used to decrease entropy inside of the little boy there. So now looking at the equation of photosynthesis, living things use solar energy to maintain life. To maintain life, you need a state of less entropy, at least a maintained amount of randomness. So what plants and producers do here is they take 6 molecules of CO2, 6 molecules of H2O, and that's pretty chaotic. That is a state of more entropy compared to, on the right, the glucose, which is just 1 molecule. I guess we have 6 molecules of oxygen there, but still only 7 total, right? 7 and 12. So on the left, there's more entropy. How can we decrease entropy? Well, we can decrease entropy in the system that is this plant by adding light energy. We add light energy, and that energy can be used to get work done. Specifically, combine all of these molecules into just, well, 1 useful molecule. Just 1. That's much less disorder. 1 molecule of glucose, way less disorder, and then these 6 molecules of oxygen, which aren't exactly part of our discussion. The light energy is stored as chemical energy in the form of glucose. So that means less entropy in the products of this reaction. Whew! Down here on the left. Animals do literally the exact opposite from plants. I'm going to erase all my scribbling here. I know some students hate it, some students like it, and on average, I guess it's just okay. So look at these two equations. I'll underline this one again. This equation and this equation, they are literally the exact opposites of each other. So the products of photosynthesis are the reactants of respiration. That's respiration down here. That's what animals or heterotrophs do. And then the products of respiration here, 6 molecules of carbon dioxide, 6 molecules of water, is exactly what the plants use. The only thing that's different is that animals on the bottom here, we produce heat energy as a product, as a waste product, and then plants use light energy. So what does this mean in terms of entropy? Well, I'd like to ask you for a moment. On the bottom here, can you remember, is it the left or the right side that has more entropy and why? Pause the video. Okay, now I'll explain it to you. Because on the right, there is more molecules, a total of 12 molecules, and also heat is being released, which tends to spread out from areas of hot to areas of comparative cold. So on the right, there's way more entropy and randomness. So about reactants and products, chemical reactions in living things. Arrow 1, chemical reactions are processes that form or break chemical bonds between atoms. So it's all about chemical bonds. Typically, we're going to be talking about covalent bonds, sometimes ionic bonds as well. Hydrogen bonds, not really at all. Chemical reactions convert reactants to products. So when we say reactants, we mean basically the ingredients. In the case of respiration, right here, those are the reactants. Convert reactants to products. Reactants and products in a chemical reaction have the same number and type of atoms. Let me explain and prove that to you using this example. I will not ask you to regurgitate this example. I simply ask that you understand the sentences up there. Reactants and products have the same number and type of atoms. So the reactants here, this C means carbon, and then it has a 6. So that means that there are a total of 6 carbons in that molecule. Then the H and the 12, that means there's 12 molecules of hydrogen, 12H. And then the oxygen, that's an oxygen atom, that means there's 6 of them. So O6, okay, great. And then let's see, 6 O2s. This is a little more complicated. So O2 means a molecule, and this molecule has 2 atoms of oxygen. So 6 times 12, 6 times 2 is how many atoms of oxygen there are in 6 molecules of oxygen. 6 times 2 is 12. So I'm going to write that down here. 12 oxygen atoms. Okay, and on the right-hand side, also a little more complicated here, 6 carbons. So that is just the same thing, 6 carbons. All right, we're already on track for carbons. You can see they're all the same, right? 6 molecules of carbon dioxide. So again, here we've got 2 atoms of oxygen in a carbon dioxide molecule, and we have 6 molecules of carbon dioxide. So 6 times 2 is 12 for oxygen. 6 times 2 is 12, so 12 oxygens down there, and that's done for CO2. Water. 6 molecules of water. So each molecule of water has 2 hydrogens. 6 times 2 is 12. 12H. Great. So again, the hydrogens have already balanced out. And then let's see, we have 6 molecules of water. Each water molecule has 1 oxygen. 6 times 1 is 6. So plus 6 oxygens. I should add commas in here. It looks like I wrote 120. Okay, so here, oxygen 6, and then 12 more 6 plus 12 is 18. 12 plus 6 is 18. So again, the same number. That was a long explanation. I apologize, but I think it will help some of you. Products also may be some form of energy. Chemical reactions. Reactions can be categorized as exergonic or endergonic based on energy gain or loss from the system, or from the products and reactants, I guess I should say. Number 1, we will look at exergonic reactions. So those release energy, so they might make you warmer, like a fire in the fireplace. The reactants contain more energy than the products in exergonic reaction. So you start a fire in your fireplace. The wood obviously had more energy in it than the ash that remains after you have burned all the wood. Example, more pertinent to biology, respiration. Once again, glucose, oxygen, those are the reactants. And the reactants have more energy than the products. So look at this diagram here. We're going to follow the blue arrow down here, and it's going down. We're kind of saying, I guess, it's just a cartoon, right? It doesn't mean anything in science, actually. But they're trying to kind of represent that the products have a lower amount of energy than these high and mighty reactants that have more energy. Where did the energy go? It was released, and specifically, it was released as heat energy, some other things as well, but we'll just say heat energy. Same thing for starting a fire in the fireplace. Wood has a lot of energy. Ash has very little energy, and where's the difference? Well, the energy was released in the form of heat. These are exergonic reactions. 2. Endergonic reactions. Endergonic means that there's going to be an input of energy, so you should immediately, well, could immediately think photosynthesis, the input of light energy to the system. So it's the products have more energy than the reactants. Products contain more energy than reactions, right? For example, in photosynthesis, we've got carbon dioxide, water, and light energy. So the reactants here are just the chemicals, right? So light isn't a reactant. We could kind of think of it as maybe a catalyst. The reactants CO2 and H2O have less energy than this product, or than this combination of products, glucose and oxygen. So in photosynthesis, we produce a high energy molecule, glucose, and then some O2, and the reactants are super low energy. If you've ever tried to see somebody gain energy by drinking water, it's not going to work. Maybe an interesting diet, but I would not recommend it. CO2 also very little energy in the bonds that connect the carbon to the two oxygens. Most energy in the products. This is kind of like lighting a fire in the fireplace, which I already said. When you burn a log, it releases a lot of energy, but the wood molecules are not moving back and forth with enough energy to cause combustion to occur. The oxygen atoms in the air required to hit the wood with enough kinetic energy, they're not moving fast enough to start combustion. So you take something like a lighter or a match, maybe a little paper, you spark the gas inside the lighter, and that increases the kinetic energy of the oxygens in the vicinity of the fire that you've started now with the lighter, increases their kinetic energy so that they ram into wood molecules with enough force to cause combustion to occur. That releases energy, causing a chain reaction, heating more molecules nearby, also causing them to hit each other with enough force to cause combustion to continue. Activation in biological systems is usually represented like this. We'll give another example of using a lighter to get something started. So on the right is a marshmallow. Is anyone hungry? Is anyone nauseous? So a flame ignites the sugar in a marshmallow. You get this flame there, and it causes the oxygen molecules in the air to run into these sugar molecules fast enough that, boom, we get combustion started. Combustion releases more energy, boom, more kinetic energy of molecules. They're ramming into each other. Combustion is going. At this point, we have reached, look at this graph here, look at the top, we have attained the activation energy. At this point, it will be self-generating. The fire generates enough heat to keep it going. So as the marshmallow burns, the products lose more and more and more energy until you're left with just a charred sort of thing on the stick. The products. Where did this energy go, right? Because energy cannot be created or destroyed. So if the reactants had all this much energy at the top, and the products have so much less energy down here, well, you could probably guess that the difference of energy was lost as heat if you just burned up the marshmallow. I, of course, would have eaten it. So the well, that's a different story. Energy unleashed. So, marathon runners tend to store glycogen, which is a polymer of glucose, in both their liver and also their muscle cells. When they need it, they are going to convert the energy in glycogen to glucose. So this is still potential energy. Glucose is a shorter term store of energy. Glucose is used to do respiration. I could add that oxygen is also used here, but it's a side note for this discussion. The waste products are carbon dioxide and water, and the only useful thing produced by respiration is ATP, or the money of the cells, and marathon runners use ATP to cause muscle contractions to occur so that they run as fast as possible. They produce heat, which is an increase in entropy. So marathon runners are essentially entropy machines. They take this very low entropy molecule, glycogen, because it's a big polymer, organized, stable, not breaking down spontaneously, and they convert this one or too many, but anyway, it's a big molecule. They convert this big molecule, glycogen, into many tiny, smaller molecules that diffuse out as you breathe them away. The water is lost as sweat on your skin, and ATP is used to do work, and in the end, it is all lost as heat, which cannot be gotten back in any way. Energy carrier molecules. So glycogen, glucose, carbon dioxide, water, none of those carry energy in a useful way in cells. The only molecule we're interested in this chapter is ATP. Skip to the bottom right here. NADH and FADH2, you can completely ignore both of those words in this chapter. They are more relevant to other topics, photosynthesis and respiration. So ignore them for now. X. Triangle number 1, food energy cannot be used directly to power energy requiring reactions. For example, you cannot use a banana to make your muscles contract. You can't smash the banana into your muscles. You cannot eat the banana whole and transport it to your muscles where it causes muscle contraction. Obviously not. You can, however, extract the energy from the banana and pass that energy to an energy carrier molecule, specifically ATP, which can then take it to the parts of your muscle cells that will use it for muscle contraction. Energy carrier molecules can act as intermediates to carry energy between exergonic and endergonic reactions. Remember that respiration is an exergonic reaction. Respiration also produces ATP. So ATP is produced by an exergonic reaction called respiration, and that molecule can then be used to do endergonic reactions. So endergonic reactions require energy and generally reduce the amount of entropy in a system. An endergonic reaction example would be active transport, DNA synthesis, protein synthesis, building muscles. That would be basically an example of protein synthesis. So ATP links respiration to all the endergonic reactions of the body. Last, energy carrier molecules are used only within cells, inside cells, because they are unstable. ATP, super unstable, super short-term storage of energy. That's why we don't say it stores energy. We say that it carries it from an exergonic reaction to an endergonic reaction. Just carries it briefly. More details on adenosine triphosphate, ATP. So triangle number 1, adenosine triphosphate is the most common energy carrying molecule. The other couple we're not going to worry about for now. It's composed of adenosine molecules and 3 phosphates. I will highlight adenosine now. Adenosine is these 2 shapes right here. The first one, this is adenine, that is also a part of DNA. You don't need to memorize that now. And then this is ribose here. That's a sugar. So there's a sort of a nucleotide here. Anyway, that's not the functional part of this molecule. The functional part is the 3 phosphates that are attached to it. So let's look at the bottom diagram here. That's B, ATP. So T means tri, 3 phosphates, 1, 2, 3. Now let's look back at the top. And the top has just two phosphates. The top diagram here, I've underlined the two phosphates twice. And then there is an independent or inorganic phosphate. That's why it is a P sub I. The I stands for inorganic, meaning it's not bound to any carbon atoms. Respiration essentially takes this third phosphate, the inorganic phosphate, and binds it to the first two phosphates that are underlined twice to the left in diagram A on the top. So an input of energy, respiration, that comes from food that we eat if we are considering an example of an animal, and the result is ATP. Why is there more energy in ATP? Well, it is a bigger molecule. It is more organized, 1, 2, 3 phosphates right in a row. And the last two phosphates are bound together with a very high energy bond. Why is it a high energy bond? That's more a topic for a chemistry class. Essentially, you can imagine each phosphate is negatively charged. Negative charges repel one another. So this third phosphate really wants to explode and break off from the first two because they're all negatively charged and same charges repel one another. So moving down to the bottom, we are going to look at how ATP is converted to ADP. D stands for di or two phosphates, only two now, and then the third has been, this third one right here has been released. It has been released when that bond is broken, releasing a huge amount of energy, and this energy can be used to get work done. How is it that work is done? Essentially, this third phosphate, and I'm going to just give you a little bit more detail than the book goes into for now because it'll probably help you understand a little bit more. This third phosphate will actually bind to something. It binds to something. Binds, you know how to spell that. Binds to something, generally a protein, and the protein can get stuff done. We've talked about active transport. This third phosphate would bind to that active transport protein, cause the protein to move or change shape, and thereby move a molecule up its concentration gradient. Remember, we talked about the definition of energy right at the beginning of this lecture. That is the ability to get work done, and work is moving something. So with this example, that third phosphate can bind to a transport protein, and then that transport protein will move something, i.e. a molecule, up its concentration gradient. Adenosine triphosphate, ATP. Triangle 1, the energy released when ATP is broken down into ADP plus inorganic phosphate is transferred to endergonic reactions through coupling. Coupling, what does that mean? Coupling means we put two things together. And what are those two things? They are, number one, an energy releasing reaction. Looking down here, this is an exergonic reaction on the left. That's an energy releasing reaction. And then on the right, this is an endergonic reaction. That's an energy requiring reaction. So endergonic reactions generally are not going to happen as spontaneously as exergonic reactions. And of course, endergonic reactions require energy. It's got to come from somewhere. So we talked about ATP right in the middle here being the source of energy for pretty much getting all work done inside of a cell. And that's where the energy comes from to get stuff done. In this example, energy from ATP is released. Looking down here, is released, and we get a release of energy, and then that energy is used to get some work done. In this case, we are talking about building proteins, protein synthesis. So taking multiple amino acids and linking them together to form a protein requires energy, and that energy comes from ATP. This is an endergonic reaction. Energy comes from ATP, i.e. the reaction takes in energy. Let me just talk about one more thing here. The protein here on the top is a higher energy molecule, and we can also consider it as having shown sort of a decrease in entropy because a protein is a polymer, a big macromolecule, and amino acids are the monomers. So amino acids are comparatively a greater amount of entropy because they are more molecules. Right. Beginning at the beginning of this diagram now, so the end point is producing a protein. Let's take a look at the top left of this diagram. It says high energy. So all the molecules to the right here, these are high energy molecules. The first one being glucose. So this first reaction here, we take a high energy molecule, i.e. food that has come from something, food energy, so from food, we get glucose and we're going to break it down, releasing energy. The products are lower energy, so CO2 and H2O, that would be respiration, the products of respiration basically. Those are lower energy molecules. You look at the bottom left of this diagram, the lower we go, the lower the energy content of the molecules. So when we break down glucose, we produce or we release energy, and that energy is very temporarily stored in high energy molecules called ATP, which have a unique ability in cells to get work done. When the ATP molecule expends its energy by breaking that last phosphate bond, we've already said this, energy is released and it is used to do work. For example, protein synthesis. The other products of the breakdown of ATP are shown down here, and they are lower energy, an inorganic phosphate and an ADP. I stated previously that essentially what respiration does is it takes these two molecules, ADP and inorganic phosphate, it takes these two molecules, adds energy from the food we eat, generally in the form of glucose, and it regenerates the cell's supply of ATP. Coupled reactions. Triangle 1. Since exergonic reactions release energy and endergonic reactions require an input of energy, your cells use exergonic reactions to help drive endergonic reactions. This is called coupling. Coupling reactions. Triangle number 3. When the product of an energy-releasing reaction fuels an energy-requiring reaction. Pause the video for now, and can you identify from that last slide that we talked about, the energy releasing reaction, what was that? And then what was the energy requiring reaction from that last slide we talked about? Pause the video for a moment. Okay, I'll now go over the answer. The energy releasing reaction was the breakdown of glucose, or respiration. The energy requiring reaction in that example was protein synthesis. Triangle number 4. Example: the burning of your car, the burning of gas in your car is an exergonic reaction, and the energy released is used to move the pistons in the engine, and that's an endergonic process requiring energy. Thereby, we get work done. The car moves. So again, adenosine triphosphate. Triangle 1, little challenge for you. Can you explain in your own words how this is an example of coupled reactions? Why is it necessary to couple endergonic and exergonic reactions? Pause the video and try to restate what we have just talked about in the last couple of slides. You can pause the video. If you have not paused it, I will now go on. So explain in your own words how this is an example of coupled reactions. Coupled reactions are when we take an exergonic reaction, we link it to an endergonic reaction, and the exergonic reaction is used to power the endergonic reaction. Second question, why is it necessary to do this? Because endergonic reactions require energy and exergonic reactions release energy. Thereby we couple them, powering the endergonic reaction. Enzymes versus plastic. To relate enzymes and energy to the wider world, enzymes is our next topic in this chapter. I'd like to use the example of marine species eating plastic and then it gets stuck in them. So this diagram, this picture down here, percentage of marine species worldwide ingesting or entangled in plastic. So sea turtles, 86% of species have ingested, that means individuals that get caught in nets or that get intentionally caught or are identified by scientists. 86% of them have some type of plastic in them that they have eaten. 86% of the species get involved in entanglement with seals, mammals generally. 23% of species with ingestion, about 28% get problems with entanglement. And then the last one, birds, you can see the numbers here, 36% and 16%. So an introduction, 18 billion tons. By the way, you don't need to memorize these terms, just general idea. 18 billion tons, i.e. that's a lot of plastic have been produced, and most of it is still with us. It's slow to break down. It's a stable molecule. 9% has been recycled and 12% of it has been burned. That leaves about 80% of it still here. 14 million tons still here. Homo sapiens are projected to produce another 43 billion tons by 2050. We have a much bigger economy now than we did at any time in the past, because that's how capitalist economies work. They get bigger and bigger. Nanoplastics. These are ground up plastic bits that are smaller than 0.001 millimeter. That is 1 micrometer. How big is 1 micrometer? That's about a quarter the size of a typical bacterium, or half the size of a typical bacterium. Bacteria range from, say, 1 to 5 micrometers. 1 to 5. 1 would be this value right here. You don't need to know those conversions, but well, I wish we all did, but this is the metric system. The rest of the world uses it. We don't. Suffice to say, that's about how big a bacterium is. That's about how big all of these ground up bits of plastic are now, because they simply get mechanically ground up by weathering, various weathering processes, whether it's the wind grinding against rocks, in the water, etc. Anyway, they're really small. Research using animals and wildlife. Is there an ethical issue there? I would say so, and there's always an ethical issue when we experiment on animals, because quite often about half of them die. Research using animals and wildlife has linked these particles to cancer, infertility, and inflammation. The source for that is right here, so if you feel like looking at it, you can type it in, take a look at it. Nanoplastics, though, I have a fair amount of experience with this stuff. I did my graduate research in Beijing, China, and it was on air pollution. And their air pollution, so I said 0.001 millimeter. How big is that? It is 1 micrometer. 1 micrometer. Write that down here. 1 micrometer. And the air quality in Beijing is generally measured as anything that is smaller than or less than 2.5 micrometers. If it's smaller than 2.5, that means that it can cross membranes in living tissues, i.e. your lungs. So when you breathe in that air pollution in Beijing, it will cross through the lungs into the blood, and research has been done there that has shown that that pollution winds up in basically every single organ of the body. And that's just particles that are smaller than 2.5 micrometers. A lot of nanoplastic is as small as 1 micrometer. And in fact, that's the definition. And nanoplastics are bits that are smaller than 1 micrometer. So that stuff can cross cell membranes. If it winds up in our food, it can cross the gastrointestinal membranes. That is still a little bit under debate, because there are some there's mucus lining most of our membranes, which definitely helps to protect us. But research, as I said, using animals and also just wildlife that we have essentially dissected after it has died, we have found that they have these particles inside of many of their organs, such as the liver, kidneys, brain. Those organs tend to pick up particles in the blood more readily than other tissues. So what we put out there, it stays out there. Plastics do not get broken down easily. This is a whale shark in this picture, just getting ready to eat a nice big plastic bag that it probably thinks is a jellyfish. Plastic entanglement of marine wildlife. It's not great. On the bottom right, that's a whale. Bottom left, that's a turtle. Thinks it's probably a jellyfish. Turtles aren't the brightest creatures in the world. This one is eating some plastic. This turtle has been wrapped up in a can holder, and so its shell has grown in a deformed way. And this is just another picture of that same thing. If a turtle grows up with plastic wrapped around it, they don't have the greatest hands, as you can see, so they have a hard time getting untangled. Enzymes versus plastic. Last little bit here. Potential solution. PET, that's basically a widely used type of plastic. We've found bacteria that will digest this plastic. So PET digesting bacteria. They have some enzymes that break down this plastic. It is completely broken down. Total breakdown of the plastic into its constituent monomers. Polyethylene digesting caterpillar larvae. So the larvae are the kind of like the infant phase of this insect, and they can eat plastic as well. Challenges. Unpredictable consequences exist of releasing organisms into the environment. So shall we just release giant cultures of bacteria and maybe hordes of caterpillar into the environment? There's unknown consequences. It's called an invasive species. We don't know how it will affect food chains. Another problem, perhaps a bigger one, is simply a lack of political will and the cost. That's one of the reasons we use plastic. It's extremely cheap to use, and we like that. Solution. Enzymes from these organisms may be extracted. First, they have to be identified, and then we can extract them or just use industrial processes to make these enzymes. Then we would industrially process these plastics, which would essentially mean mixing these enzyme things with the plastics, and then they get broken down into their constituent monomers. So how do enzymes promote biochemical reactions? We just talked about using enzymes from bacteria or maybe the enzymes in the gut of a caterpillar to break down plastics. How do enzymes promote biochemical reactions? Triangle 1, enzymes are biological catalysts. What that means is they lower the activation energy of a reaction, speeding up the reaction, and they are not used up in the reaction. So they can keep doing this process over and over and over. Enzymes are proteins with very specific 3D structure. That means the types of reactions that they can catalyze are also specific because they have a specific structure. More on that later. And this 3D structure allows it to do its catalyzing jobs. Look at this graph here. Energy content of the molecule. So we can think of that as essentially kinetic energy with a high amount of energy and then a low amount of energy. And let me add to this, could be the amount of kinetic energy, but also the amount of energy in the bond. So total energy, bond energy, and kinetic energy, meaning how fast are they moving. The reason I mentioned kinetic energy first, let's take a look at this quote. Activation energy without a catalyst. What that means is the activation energy is high. The reactants shown right here, this is how much energy they have in maybe their bonds, and they need to get way up here. So how would we get them to have more energy? Basically, we would need to raise their energy level somehow. Now, you can't introduce more bond energy without doing some type of reaction. So what can be done is the temperature is raised, just like lighting a marshmallow on fire. Once it's on fire, the reaction progresses, but it will not auto combust. So activation energy is like the lighter with the marshmallow, because the lighter makes those molecules move faster, then they run into each other with enough kinetic energy, and the reaction is catalyzed. So activation energy without a catalyst, we basically need to raise the temperature. Now, activation energy with a catalyst. That is in blue. That is in blue down here. So with a catalyst, i.e. with an enzyme. The enzyme is the catalyst. Enzyme. I'm not going to write out the whole word. You can spell it yourself. Activation energy with an enzyme is significantly lower. And so that is the question. Why is it so much lower? We'll talk about that on the next slide. Let me give you just one example over here that the book goes through also. Dissolving sugar in water has a low activation energy, and that is essentially because because water is a polar molecule and it readily breaks or disrupts the ionic bonds and the hydrogen bonds, that hold sugar molecules to one another in their solid state. Low activation energy. Reacting oxygen with sugar has a very high activation energy. Oxygen actually is going to form new bonds with the sugar molecule, covalent bonds, and produce carbon dioxide. That's combustion. And so to make those covalent bonds, more activation energy is required. Dissolving sugar in water, low activation energy, because essentially what's happening is hydrogen bonds are being disrupted, and water can do that fairly easily. So how is it that enzymes lower the activation energy for reactions? Let's start with a couple of things. So number one, this is the active site of the enzyme. I've added a little more description here. The active site, it's a pocket in the enzyme, a concave section in the enzyme. It is complementary to the substrate based on its structure and also chemical nature of the amino acids in the active site, meaning are the amino acids in the enzyme charged, polar, nonpolar, etc. So it is complementary. What does complementary mean? It doesn't mean that the active site is the same shape. It means it is complementary, sort of like your shoe is complementary to your foot. So remember, your shoe is not the same shape as your foot. Your foot is firstly not hollow, and your foot cannot have another foot put inside of it. Your shoe can have your foot put inside of it. That is, they're complementary. A lock and a key are complementary. A key and a key are the same. Okay, so that's why the substrate molecules or the reactants will fit inside of the enzyme's active site. What do we mean by they are complementary in terms of their chemical makeup? Let me give you a simple example. Charged. So this last one right here, charged. If the amino acids that surround the active site in this enzyme, remember enzymes are proteins and proteins are polymers of amino acids. If they are all negatively charged, so here's a bunch of negatively charged amino acids in the active site, I'll make some circles around them. So a bunch of negative charges. What that would mean is the substrates must be complementary to the charge in the active site. So you can pause the video and think about it for a moment. Would these substrate molecules likely be positively charged or negatively charged? Okay, if you haven't paused it, you missed your chance. These substrates would likely be positively charged because positive charges are attracted to negative charges. So the substrate's chemical nature is also complementary to the active site. So step 2 down here. The substrate and active site change shape, promoting a reaction between the substrates. They kind of missed one part, which is, of course, the substrate molecule enters the active site. I guess it says that here. The substrate molecules enter the active site. Now, let me say this. When they have done that, it is not a perfect fit. It's like you putting on a shoe that is somewhat too small for you. So what that does is it stresses the substrate molecules. It stresses their bonds. Like a shoe that is too small, it stresses your foot as well as the shoe. Temporary bonds are formed between the active site and the substrate. The bonds in the substrate molecules are stressed similarly as your foot is stressed when it's put in a shoe that is too small. This is where the lowered activation energy state is produced from. Activation energy is lowered because instead of increasing the temperature of the entire environment this reaction is happening in, we simply have this almost perfect fit between the active site and substrate. So when the substrates move into the active site, they are stressed in a specific way that catalyzes the reaction, thus producing, lastly over here, our final end product here. Okay. Last thing here you want to note, number 3, the substrates bonded together, they leave the enzyme, so now they're a product, and the enzyme is ready for a new set of substrates. So this would happen again, and this can happen really fast, depends on the enzyme, depends on the substrate. 10,000 molecules or catalyzations or reactions per second would not be uncommon, 20, 30, 40,000 per second. So this is really, really fast. Enzymes and reactions. The sum of all the chemical reactions inside a cell is called metabolism. Many cellular reactions are linked through metabolic pathways shown here. These pathways are generally catalyzed by enzymes, not always, but most of the time. So initial reactant is shown right here. This would be like a substrate. We could also call it a substrate. The substrates are essentially the reactants when an enzyme is involved. Then we have enzyme 1 that produces a second substrate. That is substrate number 2, B. Now, we just talked about in the last slide that enzymes produce products. So is B a product or a substrate? Well, fine. It is the product of enzyme 1, but then enzyme number 2 is going to treat that product as a substrate and then produce yet another molecule that we would say is the product of enzyme 2. So C is the product of enzyme 2, but then C is also the substrate of enzyme 3. Notice here also that C is also the substrate of enzyme 5. Now, you can kind of think of this as an assembly line in a factory. One thing produces the next, and then the next worker takes that and improves on it or changes it a little bit. These are metabolic pathways. How are enzymes regulated? So enzymatic reactions can change based on concentration of the enzyme and or substrate, but this is not always feasible within cells. So let me say, why is it that enzyme reactions can change based on the concentration of the enzyme or the substrate? So you can think of enzymes as workers at an assembly line or maybe workers building a house. If you add more workers, then you're going to get more work done. Pretty simple. Substrate molecules, you can sort of think of them as the nails that are used to nail a house together. If the workers are always running short of nails, then they will not have anything to do. So if you add more substrate, i.e. add more nails to the work site, then those workers, the enzymes, will get more work done. Okay, so the problem with this is that cells can't always just add more substrate because they may not have any. It may be that that substrate is oxygen and the organism's already breathing as hard as it can. It also may be that they cannot produce enzymes that rapidly. Triangle number 2, there are a few other ways a cell can regulate enzymes within themselves. Triangle number 1, controlling enzyme synthesis by turning genes on or off. So let me remind you of the general nature of information in a cell. DNA is where genes are coded for. There's this mRNA molecule. It basically carries the information in genes out of the cell to ribosomes where proteins are made. So if a gene is on, then we are going to get protein synthesis. Yay, big smiley face. However, if, for example, we have maybe the protein in this case is an enzyme and it's producing too much substrate, we don't need any more of this substrate, maybe it's even at a toxic level in the cell, what we can do is turn the gene off. When we turn the gene off, no mRNA is produced and therefore no protein is produced either, and the concentration of this enzyme in the cell goes down. If there's less enzyme, then there's less of this reaction and less product produced. Genes regulate metabolic pathways by controlling enzyme synthesis and activity. Just mentioned how we can control enzyme synthesis. That is by turning genes on and off. Triangle number 1 is about something else. Competitive inhibition. Drugs and poisons often inhibit enzymes. Inhibit means to slow down. Inhibit enzymes by competing with a natural substrate for the active site. Let's take a look down here in this diagram. On the left, this is just a typical enzyme in purple here with its active site. You can see that the blue molecule is complementary to the active site, so it'll fit in there. Now, a competitive inhibitor, let me bring your attention back to this triangle number 2 here. Competitive inhibitor molecules are almost always very similar in structure to the substrate of the enzymes, which enables them to bind to the active site. So in B here on the right, you can see that in the yellow molecule that is a competitive inhibitor. Now, it's not the same shape as the substrate. If it was the same exact structure, then it would be the substrate. It's a similar shape, which allows it to bind to the active site. If there is this inhibitor molecule in the active site, then the substrate molecule cannot bind to it. It would be kind of like taking a key and breaking it off in the lock. You have essentially ruined that lock until the blockage is removed. Competitive inhibitor molecules bind reversibly. Let me erase some of these scribbles here. They bind reversibly. So this competitive inhibitor molecule will likely bounce out of there eventually. How soon? Could be a microsecond. It could be 10 seconds. It depends on the inhibitor and the enzyme. So what that means is that triangle number 3 here, the greater the concentration of the inhibitor, the more inhibition we have. So if there's only, say, one inhibitor molecule present on this whole in this whole beaker here that I'm now drawing, and this inhibitor molecule unbinds to the active site, then it may be some time before it binds again to the active site. But if there is a high concentration of competitive inhibitors just bouncing around everywhere nearby, then as soon as one inhibitor molecule leaves the active site, another one is more likely to enter it instead of a substrate molecule. So increase the concentration of the inhibitor and you will increase how much inhibition you have. You'll slow the reaction down more. So this is why it is called competitive inhibition. The substrate and the inhibitor molecule both compete for the active site. Non-competitive inhibition. So triangle number 1 here, non-competitive, that means the inhibitor molecule is not competing for the active site with the substrate. Something different. A molecule binds to a site on the enzyme outside of the active site. Where is that? Let's take a look in the bottom left diagram here. This is a site where a non-competitive inhibitor might bind. Binding of the non-competitive inhibitor. This causes, direct your attention up here, this causes the enzyme's active site to change shape. So it can no longer bind to its substrate. So binding of the active site to its substrate is completely shut down. Take a look bottom right here, C. We now have the non-competitive inhibitor bound to this other site outside of the active enzyme, and the active site has changed shape. No longer is it complementary to the substrate. The substrate cannot enter the active site. This would be sort of like you trying to open your mouth to take in some food, except somebody keeps jabbing you in the ribs, causing you to wince and close your mouth so you can't eat it. Maybe that's a useful analogy for you. Triangle number 2 here, binding of the inhibitor molecules is generally reversible. It is less reversible than competitive inhibition, though. So it's generally reversible. That is acceptable for the exam. Books really like to say it's not reversible, but the fact of the matter is, it is reversible. It is just much less reversible. So this is a fine thing to say for the exam, even if your book says it slightly differently. Lactose intolerance. There's a great video on Blackboard, or a link to it on Blackboard, and it's about 15 minutes long, really interesting. It is optional, so those of you who are pressed for time, it's not required. It's simply an interesting video that might help you to understand this. So lactose intolerance is as follows. Babies produce lactase throughout early childhood. Lactase is an enzyme that breaks down lactose. So this is an enzyme. So if you produce this enzyme, you can break down lactose, the main sugar in breast milk. So babies obviously produce it because they breastfeed, they eat or drink milk. As we grow up, many people stop producing lactase. These people are lactose intolerant, meaning they can't digest it. They've stopped producing the enzyme to break it down, so they can't digest it anymore, and it causes all these problems. Indigestion, fatigue, flatulence, etc. It's not great. So why is this? Meaning, why would people stop producing lactase, whereas some other people continue to produce it? It essentially comes down to natural selection, and that video explains this in more detail. In natural selection, early humans had no access to milk after breastfeeding. So early humans, before we domesticated cattle, sheep, and gained access to animal milk. Adult humans do not feed on breast milk, at least not normally. Early humans had no access to milk after breastfeeding. So producing the enzyme after childhood was a waste of energy. And over long periods of time, thousands of years, tens of thousands of years, individuals whose cells are wasting energy producing an enzyme that does nothing, because if the person is not drinking milk, then they have no access to lactose. That's the only place lactose really occurs. It's a waste of energy. So over long periods of time, those individuals were less fit because they were wasting energy doing things that producing enzymes that did not contribute to their survival and reproduction, and thus passing on of those genes. Lactose intolerance varies by geographic region. Sorry, this should be region. Most common in East Asia, West Africa, and also Native Americans or those with Native American ancestry. In the Middle East and Northern Europe, where milk-producing livestock was domesticated, mutations coding for the continued production of lactase, the enzyme that breaks it down, provided a selective advantage because milk is an excellent food source. So over very long periods of time, having these domesticated livestock who would just stand there and let us get gallon upon gallon of milk from their udders gave human beings a tremendous food source. Milk, by the way, is the only food on earth that wants to be eaten. All right? Cows do not want to be eaten. Plants do not want to be eaten. That's a topic for that's another topic, but they produce all kinds of compounds that are an attempt to discourage animals from eating them. So milk is designed to have everything a person needs to grow. That's why babies can survive on just breastfeeding, no problem. It's designed to have everything you need. So anybody who had a mutation that enabled them to gain access to this tremendous food source, they were more likely to survive and reproduce and pass on those mutations to successive generations. These mutations gradually spread east, west, north, and south. Okay, lastly, about lactose intolerance, before we move on, I'd like to mention I worked in China for a few years, and you look this up online, it says something like 90% of people in East Asia, especially Northeast Asia, are lactose intolerant. Everybody eats ice cream and drinks milk in where I was. This was Beijing, China. And the reason that they have no problems doing that is that we have been able to isolate the enzyme that breaks down lactose. So we can put that enzyme, essentially put it into milk. It breaks down all of the lactose into its constituent monomers, which everybody can break down. So that's why even if you are lactose intolerant, milk can be made that doesn't have lactose in it. Okay, environmental conditions affect the activity of enzymes. They can break the enzymes, speed them up, or slow them down. Enzymes only work because the three-dimensional shape of their active sites can fit the shape of the substrate. If an enzyme loses its three-dimensional shape, we say it has been denatured, and the enzyme will no longer function correctly anyway. Generally, denaturation occurs when bonds, mostly H bonds, are disrupted. The reason for that is hydrogen bonds contribute significantly to holding the tertiary and also quaternary structure of enzymes together. Other bonds are also there, but if even the hydrogen bonds are disrupted, the enzyme's shape is altered. Perhaps not completely destroyed, but altered. And H bonds are also the weakest type of bonds. Ionic bonds, disulfide bridges, covalent bonds, those are stronger. So the H bonds are the first to go, and when they go, typically the enzyme's shape will be disrupted enough so that it does not work anymore. Enzyme shape can be disrupted and denatured by many environmental factors, including pH, salts, temperature, and the presence of coenzymes. So effects of pH. Enzymes are denatured and lose their function when pH is too high or too low. So for example, in the stomach, we have enzymes that are produced and designed with a structure that is adapted to a pH of 2. So enzymes in the stomach will only function at or near a pH of 2. Looking at the graph down here, let's take a look at this fella, pepsin. That is an enzyme that breaks down proteins. Its maximum activity occurs at about pH 2. It can function at higher and lower pHs, but not that much higher. If we get to, say, pH of 4 or 4.5, then no catalyzation of reactions is possible for that enzyme. Let me make a note that you can go back to an earlier chapter and take a look at protein denaturation, and that might help you to understand what's going on. For trypsin, this one is in the intestine. So in the intestines, the pH is higher, around 8. And let me circle this one. It says for trypsin, that's also an enzyme that breaks down proteins. The maximum activity occurs around pH of 8. And so right here, maximum rate of reaction catalyzations. And if we get above that too much, the rate goes down. If we get way above it, then the reaction completely stops. So at 10, no reactions can be catalyzed whatsoever. Let me give you just a little bit more detail on what you could be thinking about when an enzyme is denatured. So enzymes are usually like a 3D structure, and they're always drawn like sort of a blob. They are technically called globular enzymes. So here's a globular enzyme right here. Now it's a big blob, right? But within that, remember, it's an enzyme, so it is going to be essentially a polymer of amino acids. So I've drawn a circle and a square. These are different amino acids. And these amino acids have to be essentially connected to each other, folded in just the right way so that the enzyme can perform whatever function it is that it performs. In this case, it's an enzyme. So I'll draw a heart for the last one. So this is a long string. Now, let me add one final thing here to hopefully this won't get too messy. So remember, an enzyme has a three-dimensional structure. We could say it is sort of a globular protein. But remember, a protein is a polymer of amino acids. So there's this string or polymer, a string of amino acids held together by all kinds of bonds. And if any of those bonds are disrupted, then this string will unravel. So instead of being a globular enzyme, when denaturation occurs, this string of amino acids will then start to look something like this. So that's still a polymer of all those amino acids, right? So there'd be one amino acid here, then it'd be connected to a square, then a triangle. These are all just different amino acids, etc. I won't redraw them all. This enzyme is totally denatured. It has lost its tertiary or quaternary structure, or both. Its active site has been totally altered, totally destroyed, and so it can no longer catalyze reactions. Effects of temperature. Effects of temperature. Low temperatures slow down molecular movement. So that means that the active site and substrate molecules will collide less and also collide with less energy. So it's a little less likely that a reaction will be catalyzed. High temperatures increase molecular movement and increase the speed of the reaction, but only to a certain point. Take a look down here at the graph. Temperature is on the x-axis, and as temperature increases, the rate of reaction also increases up to a point. This is called the optimum temperature for an enzyme. That's where it is working as fast as it can. But if you increase the temperature beyond this point, and note for humans, that is the optimum temperature is about body temperature, 98.6 degrees Fahrenheit. If we increase it too high, this triangle here, when it gets too high, it causes the enzyme's shape to be altered, destroying its function. So that means you can think of, remember that chain of amino acids. It's like it's a polymer. It has been wound up like a ball of thread, and when you start shaking this ball of thread enough, that ball of thread will unwind, i.e., the protein will denature. So shake it too much, which is what happens when you increase the temperature, right? More movement, kinetic energy of the molecule, and it just isn't stable at that high of a temperature anymore. So this is why a person with a very high fever, say 108 degrees or higher, can suffer serious damage to their body and especially to their brain, because enzymes stop working correctly. They will catalyze reactions not as effectively, or some may catalyze them too rapidly even. Point being, fever is bad because it changes the rate of reactions for enzymes and also metabolic pathways. So metabolic pathways. Enzymes are regulated, and that's how we regulate metabolic pathways. Temperatures affect the rates of reactions. Inhibitors affect them. Gene expression, turning on genes, turning off genes affects the amount of enzymes. That also affects these metabolic pathways. So they are carefully regulated and are sensitive to changes. So last triangle here, how might the end products E and G be affected if a competitive inhibitor for enzyme 3 were added? So the end products, let me direct your attention here, end products E and G, how would they be affected if we added a competitive inhibitor for enzyme 3? This is a really tricky question. You will need to pause the video and review what we have so far talked about before you can answer how a competitive inhibitor for enzyme 3 might affect the amount of end products, E and G, for this metabolic pathway. Pause the video and think about it.